Hello everyone. In this part of presentation, we will discuss the quantitative measurement of bacterial growth. The various principles and methods used for the cell quantitation or the bacterial growth assessment will be discussed. All of you may know that bacteria is a single cellular organism. It's a unicellular organism. So the growth of a bacteria is usually refers to the number rather than their size and other features like in a multicellular organism. So the number of bacteria is important, and the change in number is usually reflecting the growth or reflected as the growth of a bacterial system. So we have discussed about the reproduction system in bacteria, like they will be doing the binary fission mostly and some other mode of reproduction, but usually they prefer the binary fission like mode of reproduction, or simply they are doing this process by means of asexual methods. So we are moving into the content or into the various methods, and before that let's see the two common terms we have to face across the presentation is total count and viable count. The count or the number of bacteria, you can take the number of bacteria by means of different methods, but when you take the number could be of either a total count or it could be a viable count, right? So the total count is simply refers to the total number of bacteria present in a media, in a broth or in a system, irrespective of their living status. That means the total count include the count of bacteria including both the living and dead cells. This is because there are certain techniques, mostly the microscopic techniques like staining and microscopy, and some other machine based counting techniques like electronic counters, and such methods will rely on the particle. That means they do not check whether the cell is live or not. So in that situation, usually we will get the number as a total count. But when we are going for the methods like serial dilution and plating or membrane filtration and plating, and most of the plating experiment or most of the culture based experiments will give you a number which is only of the living organisms, or those cells which could multiply only will be giving you a count. So such counts are called, or such numbers we obtained are called viable count. All right. So additionally, recently we have advanced instruments like a flow cytometer or the FACS, that means the fluorescent assisted cell sorter, like machines are there which could differentiate the dead cells and the viable cells separately, and you will get the count of both separately. This is the overview of various methods usually were performed in laboratories for the cell quantitation methods, and few of them are commonly executed in laboratories, and many others are usually performed for or only for research purposes, right? And what will be the requirement, what will be the need of doing this one? There are three major concepts or major principles under which we perform this quantitation, right? In other words, we have three principles of cell quantitation methods. One is cell number, then second one is cell mass, and third one is cell activity. So we can take the cell growth or the bacterial growth by means of either cell number, or by means of cell mass, or by means of cell activity. Apart from these three concepts, we also have another mode of classification like direct and indirect measurement. So in this slide you can see certain methods are listed under direct, which is actually I have given a green color, and the other methods in the light blue color. So those in the green color are representing the direct methods. A direct method is actually representing, or these are the techniques in which the number of the cells are directly taken. All right. You can have a lot of examples. There you can see the counting chamber, which will give you a number directly from the chamber. Similarly, for the flow cytometry, the breeds count, or even the dry weight. In case of dry weight, the weight of the cell is directly taken. So, but in case of the other one, that means the indirect methods, in case of indirect methods, we will get the number in an indirect way. That means the result of a particular test will not give you directly the number, but instead from the result obtained from the test, we have to find out the number. So that's usually happening in case of experiments like plating experiments, or in case of turbidity analysis, or other analysis like cell activity based methods. So in all those experiments, we will get a result. For example, if you get the colonies in a plate, the colonies represents a cell, but it's not like a colony is a single cell. We know that a colony might have contained thousands to lakhs of cells. So a colony but represents a cell. So in that way, when you get a particular colony, or when you have 10 colonies, we will indirectly say that there are 10 bacteria in the original sample. So that is the way we correlate the indirect methods to the number. Okay, so based on cell number, we have a number of methods in the direct concept, and also under indirect methods also we have certain methods. So and also in the cell mass we have dry weight determination and other experiments. So in cell activity also we have some experiments which are only in the indirect category. So in cell number, or the quantitation based on cell number usually relies on taking the number, right? It's based on the number the estimation is done. And in the other one, this in case of cell mass, the weight or the mass of the cell is the basis of measurement. And in case of cell activity, the activity of a cell, or the performance of a cell, or the metabolic reaction of a cell, the enzymatic reaction of a cell, all those are the basis of estimation. So first we will have to see the cell based on cell number, which are the methods. So these are the listed methods. Under direct, we have counting chamber, electronic counters, flow cytometry. These three are actually we will get or we have to use separate machines. And the other three under the direct are proportional count, breeds count, and membrane filtration, followed by staining. So these are under the direct method, in which we will use in case of counting chamber, we use a chamber, a glass like chamber, and also along with that we will also use microscopy. In the other three, that means in the proportional count, breeds count, and membrane filtration followed by staining, they are purely depending on microscopy, whereas electronic counter and flow cytometry depends on machines. Okay, and under the indirect category, the membrane filtration and plating will give you an indirect number. That means all these are purely based on plating. That means the serial dilution and plating, membrane filtration and plating, and the third one, MPN method also will give you an indirect measurement. That's a tube based method. So we'll see each of them in detail. First we'll take an example of a method which is called a Petroff-Hausser counting chamber. This is a Petroff-Hausser chamber is an example for a classical example for counting chamber used for estimation of bacterial cells. We know that these kind of counting chambers are widely used in counting of blood cells and other cells like the semen cells and all. And for bacterial counting also we can use these kind of counting chambers. Okay. So these are, like as you see in the image, these are nothing but a square marked glass slides. These are nothing but glass slides. Okay. Usually the cells in liquid cultures are counted in a microscope with the help of these specialized glass slides, and these glass slides along with a particular cover slip is usually called the counting chamber. So as I already said, this chamber has precisely machined chambers and cover slip so that cells in very small volumes can be counted using the microscope. And for the gradings, for the ruling, usually the new improved NOVA ruling pattern is followed. Okay, and the squares marked within the chamber has usually has various stages. That means there are three types of squares available. One is the large square having a dimension of 1 by 1 mm, and there will be 9 such large squares. There will be 25 medium squares within this one, within this large 9 squares, and even within each of the 25 medium squares there will be 16 small squares. So this will be the pattern, and usually the depth of the chamber is 0.02 millimeter. This image will give you an idea about the counting chamber. You can see where these square markings are found and where you have to load the cultures. And after the culture is loaded, usually a very small volume around 10 microliter is placed in the well, and after that we will wait for some time to settle the samples, to move the samples into the chamber region or into the marked region. And after that we will observe under the high power microscope or low power microscope. Usually we do not prefer an oil immersion objective, right? And this will be the appearance. In the image you can see that, and the volume of sample in the marked region, right? The volume of the sample in that particular square marked region will be calculated, and the number of bacteria present in that area also will be counted through the microscope. And after that, after counting the number of bacteria present in a particular area, the volume present in that area also will be calculated, and after that we will calculate, or we can correlate the number of bacteria present in the original sample, or per ml what number of bacteria present will be calculated. Okay. So the volume under this area will be simply calculated using the formula depth of the square into height of the square into height of the sample. Okay, that means this height of the sample represents the volume. Okay, the size of the sample, what volume of the sample is there. So this will be calculated accordingly. An example is given there. If you are using a 0.2 mm square, that means if you are using a 0.2 mm square, then the size, the depth versus height will be 0.2 into 0.2, and the height of the sample, or the depth of this height actually represents the height usually will be given on the chamber. What is the value of the height of the sample? So after that you can calculate this volume, or after getting that volume you can calculate the total number of bacteria present in that volume, and from there you can calculate the total number of bacteria. Or otherwise you can also do another type of calculation given as given here. You can use this following formula, like the number of cells per ml is equal to number of cells counted per divided or number of cells counted per square into number of squares into dilution, if any dilution is used, into 50,000. And 50,000 is a constant factor that is obtained by taking the values provided. That means 50, which is representing the inverse value of depth of this particular square, and that's converted into the cubic mm. That means that you will take multiply this value with 1000, so that you will get 50,000 as a constant value for 1 ml if you want to calculate per ml, right. So after that you will get a particular number in a square. If you are taking, you can take any square for counting. That means already we have said that there are 9 square, 25 square, 16 square. So any of the square will be taken. Okay. So if you take the number of bacteria, or if you count the number of bacteria in the 25 square, then that value will be multiplied. Okay. So it is like 25. If the number of bacteria is 28, then the number of cells per ml will be 28 into 25 into 50,000. So you will get a particular value, and that value represents the number of cells per ml. This also represents the procedure and the measurement strategy under the Petroff-Hausser counting chamber. So hope you can follow this method for counting of bacterial cells.